Okay, hey everyone, it's the matter and energy processing Prezi, also known as a tour of the cell part two. Now, in the structure and support Prezis, we look just mainly at that. What holds the cell together, sort of in place, what acts as a barrier, and gives it that support that we see. So we looked at the cytoskeleton, we looked at the cell membrane a lot, and we looked at the extracellular matrix. Now, what we're going to do is look at kind of what makes it tick. And so what's actually going on, not just the structure and support, but there's a lot happening, and we're going to focus on the matter and energy processing. And so let's go to big screen. Big questions here is that, first of all, you need to know the relationship between matter and energy. They're not the same thing, but they are connected. Why life requires matter, why does it require energy, and how do the interactions of the cell help with both of these? So, reminder of the life of the cell. All cells must do these things to stay alive. They must process matter, process energy, and process information. So, when we say process matter, we need to somehow get it. We need to synthesize it, and we need to digest it. So, we are getting things, we're making things, and we're digesting things. Okay, so acquired, synthesized, digested, breaking it down. Getting, making, breaking down. Okay. We also need to process energy. So to do all of these things, getting it, making it, and breaking it down, we need energy. And this energy usually comes from one of two places. And so where do you think that this energy comes from based on your general bio background? Okay, in a little bit, we'll see if it's the same as what you're thinking. And we must be able to process information. So instructions that enable the cell to process how to do all this stuff, it must be interpreted by the cellular system. So we not only have information within the cell on how to process matter and process energy, but we also have signals from the environment that come into play. So it's a pretty complex process. Cells also need to be able to do the following: reproduce and communicate. And so these should be something that you've seen already, characteristics of life. They need to be able to reproduce and communicate. So that information on how to process matter and energy, it must be able to be transmitted to new generations of cells. And we need to be able to commute and respond to or direct other cells that are close by. And so each cell is not in its own little vacuum of space. They're complex interacting cells structures. Let's call it a structure. So cells have systems to do all these things. So we're going to kind of look at the systems approach here. So why do cells need energy? They need to be able to do work. They need to work to get the matter. They need to work to put things together. They need to work to break things down. This energy to do all this is stored in chemical bonds. Okay, and so we're allowed to do this with the energy stored in the bonds. So when we're saying get things, put things together, break it down, this is a little bit more of an explanation. So we take in nutrients. When we say get things, we're taking in nutrients. When we're saying breaking down, we're breaking molecules down. So we're looking at like dehydration reactions. Remember that? Not dehydration, sorry, hydrolysis reactions, when we take water to cleave a bigger molecule and break it into smaller one. We build molecules with dehydration reactions. You've seen that one. There's other ways too. We make ATP. ATP, yeah, you know me. I'm going to say this all year, so get used to it. ATP is fun. ATP is necessary. ATP is going to be breathed and lived in this classroom. We need to remove waste, get rid of the yucky stuff, reproduce, and regulate everything that's going on. Now, energy is processed. Remember law of thermodynamics. You cannot create or destroy energy. It's not made. It's just processed so we can use it, okay? It does come from somewhere, the sun, originally, okay? It's just how it's packaged and how it's going to be used within the cell itself. So two major sources of energy, food and the sun. Now the sun is actually the primary source because any of the food for the cell eventually got its energy from the sun to begin with. And cells convert energy to forms that they can use using two major organelles. We have our powerhouse mitochondria and we have our chloroplast. And so we'll spend a lot of time talking about cellular respiration and photosynthesis, and see how it connects to energy formation and energy transformation. Okay, so we'll go through this a little bit fast because I just went through everything. This will become very, very comfortable. Right now, I'm guessing you have a kind of working knowledge that chloroplasts take in sun and they release sugar and oxygen. So mitochondria takes that oxygen and sugar, creates heat, well, transforms. Some heat is transformed and lost, but others converted into usable forms of energy, releases carbon dioxide and water in the process. Now, there's other organelles besides the chloroplast and the mitochondria that are involved in this whole matter processing, though. One of them is the lysosome. The lysosome is basically the area for breakdown and waste. It's a sac full of digestive enzymes. Okay. They're often kind of look like little vacuoles because, in a way, they're like vacuoles. They store stuff, but then they're going to break it down. They digest large molecules, and they also digest damaged cell parts. They play a main function in apoptosis, which is programmed cell death. And so when we look at cell division and we look at mitosis and the cell cycle, and we talk about apoptosis, it's the lysosome that's going to play a role in breaking it down. So if there's an issue with the lysosome being able to break things down, then things get backed up in the cell and other issues arise. Now, the cytosol pH is 7, which is sort of a neutral value. The lysosomal enzymes work best at a pH 5, at a more acidic pH. And the reason is because it's then going to, this acidic pH is going to be able to denature and change the structure around of some of the other molecules that are there. Okay, so it says, see if you can guess which of these two white blood cells has gone apoptotic. Okay, and so hopefully you have guessed right here, we're breaking down into cell death. Hint, apoptotic cells break apart into vesicles called blebs. You don't need to know that the term is called blebs, but you can see it when it starts to break it apart here. Oops, I'm using my finger, not the mouse. That little blobs kind of come off. Okay, apoptosis is responsible for the space between your fingers, your nostrils, your eyelids, and generally every hole in your body. So we always think of it as cell death, and when there's an issue with the cell cycle, we talk about an issue with apoptosis and cancer, but it's necessary for some of our body structures. Now, lysosomal diseases are often fatal. The digestive enzymes don't work, and everything builds up. Lysosomal storage diseases pick up molecules, but they can't digest them. If you look at ALD, which is adrenoleukodystrophy, what Lorenzo had, it was a lysosomal issue. It could not break down the fatty acids. Tay-Sachs is another one. It's a recessive genetic disease. The undigested fats build up in brain cells. As a result, you have mental retardation and death at an early age, often by 4. Now, there's other somes, organelles that end in some, and just remember that some means the breakdown of some sort. Peroxisomes is the other common one you'll come across. It breaks down fatty acids and sugars, detoxifies cells, detoxifies alcohol and other poisons. It's to help keep your body cleansed of toxins, and so you don't have that buildup, especially when it comes to like peroxides and gases that might form from that. So liver cells oftentimes have quite a few peroxisomes in them, as that is an organelle an organ that is primarily designed for the purpose of cleansing toxins out of the body. So lysosome, peroxisome, some breaks things down, gets rid of it. Vacuoles store things. It's a structure. It's a membranous sac full of storage materials. We have food vacuoles contain undigested food. Now, they will fuse with the lysosomes to help break down that undigested food. We have contractile vacuoles. In freshwater protists, they act to pump out extra water, and this helps keep the balance, the osmotic balance, in place. Plants have central vacuoles for storage, stockpiling proteins, inorganic ions. We deposit things. It's a little bit of everything, and this helps plants have the necessary forms of matter that they need in times of like drought, for instance, when maybe they can't get them. And also it helps maintain that structure of the plant cell. So we have vacuole. And animal cells, they're going to be much smaller here. And so more like these little vacuoles here. Okay. Now the mitochondria, we all know little mito, the powerhouse. It's an outer membrane. It has an outer membrane just like the chloroplast does. So basically it is a double membraned organelle. It has a highly folded inner membrane, and this allows for more surface area. So if you haven't done the surface area lab, we'll look at it a little bit more in compartmentalization. I think there might actually be a Prezi on it. And so it will answer this question of why we want all these folds, why it's good to have all these folds. Not that we want them, but that it has evolved to have all these folds. The cristae are folds of the inner membrane. So each of these folds are referred to as the cristae. And the matrix is the inner fluid-filled inner cavity. Now, the whole function is aerobic cellular respiration. It takes molecules, breaks them down, so it's a form of digestion, to produce ATP, carbon dioxide, and water. So you take these large carbohydrates, macromolecules, and you produce smaller molecules as a result. Now, since it is being broken down, it's a form of digestion. And catabolism is a fancy word that means breaking down molecules. And I think I might have mentioned in another Prezi, the way that I think of the difference between catabolism and anabolism is that cats break apart furniture with their claws. They claw at the furniture and breaks that furniture down. At least my couches from my cats do. And so I think that's how I remember that catabolism means to break apart, to break down. Almost all eukaryotic cells have mitochondria. The more active the cell, the more mitochondria. So think about it, take a minute, and think about which type of cells are going to have the most mitochondria. And hopefully you answered muscle cells. Muscle cells are a great example of types of cells that are going to be more active and need more mitochondria. Now, mitochondria reproduce independently of the cell. And this whole reproduction capability, that's another sign of the whole, another piece of evidence that might support the theory of endosymbiosis. So thanks, mitochondria, for all of your work. Chloroplast, again, like the mitochondria, it's double membrane. Outer membrane, we have an inner membrane. Now, the inside is called the stroma. Contains DNA, because remember the chloroplast and mitochondria contain their own type of DNA, and ribosomes and enzymes. It's where sugar is made. These little stacks here are called thylakoids, and this is where the ATP is made. And when I say ATP is made, it's not made from nothing. It's converted from the solar energy that comes in, and we'll look at it in photosynthesis. So in this kind of reaction, we are building bigger molecules. We're taking ATP, carbon dioxide, which is pretty small, and we're taking light photons, and we're producing oxygen and waste. Okay, chloroplasts make plants green. Why is that? Okay, and hopefully, if you haven't done it yet, we'll look at the chlorophyll and about the light spectrum and about which lights are absorbed, and green is what is actually reflected. Green is the pigment that is reflected, and so that's why they seem green. Only plant cells have chloroplasts. And so here's cells from duckweed. It's colorized from an electron microscopic view, but it's colorized so you can see the green chloroplasts here. And just so you guys know, you see your nucleus here. And if you remember, inside the nucleus, we have what's called a nucleolus, and that's going to come into play when we look at cell division. So mitochondria and chloroplasts, they have similarities. They both transform energy. They both generate ATP, believe it or not. We talk about mostly the mitochondria making the energy, but they can make some too because they're transforming the solar energy into ATP. They both have double membranes. They both are semi-autonomous, meaning they are not part of the endomembrane system that the rest of the cell forms. They both move, change shape, divide on their own, and they both have small internal ribosomes, circular DNA, and make their own proteins. So how can we explain that? And that's that theory of endosymbiosis. So this is all of the previous statements are supporting statements, supporting evidence of the theory of endosymbiosis in the formation of eukaryotic cells. And in eukaryotic cells, the idea here is that we have a cell with a nucleus. Okay, so we have like a little basic ancestor cell that was engulfed by another cell. Okay, and the mitochondria came from the engulfing of oxygen-using non-photosynthetic prokaryotes meaning before eukaryotes. And then the chloroplast was absorbed and it was photosynthetic. And so when it was absorbed, it was a positive relationship. It helped the survival of the cells, and this process was able to be transferred down in the information in the DNA of the cell and carried on to the next cells. But wait, there's more. We talked about chloroplasts, but they're just one group of plant organelles called plastids. All plastids are products of endosymbiosis. So we hear about mitochondria and chloroplasts as evidence of endosymbiosis, but there's other plastids too. And in each of these, it's a processing, plastids are processing energy or processing products to create something else. And so we have chromoplasts, which is the pigments for flowers and fruit. We have leucoplast, which stores lipids. Amyloplast, which stores starch, etc. There's similar patterns of plastid evolution and diversification as seen in algae too. And so basically all this part is telling us is that there is different lines of this occurring, instances of this occurring that lead to different organisms that have these similar structures today. And it's just kind of, if you trace it back, it shows how far back or how common ancestors are by how many times this diverged. Any questions? Just make sure you ask.